Chapter 3, The Fundamental Unit of Life. In this chapter, we'll talk about how cells are the fundamental unit of life. We'll compare plant cells to animal cells. We'll take a look at the membranes and to see what their structure is like and the function. We'll also discuss the nucleus and the organelles within a cell. Then we'll take a look at special organelles, the chloroplasts and the mitochondria and other organelles. So it says, are all living things made of cells? And you already know the answer. It's heck yes. They're all living, all life is made out of cells. And that's one of the fundamental characteristics of life. Some living organisms may have one cell and some may have trillions of cells. So in this diagram here, we see a single cell up at the top and then we see an elephant down below. But I don't need to look at any diagram. I can just think about myself and I know that I'm made out of trillions and trillions of cells. Remember, cells are the fundamental units of life. Cells can be grouped into two categories, prokaryotic cells and eukaryotic cells. Prokaryote, pro means before and kary means kernel, like a nucleus. So this means before kernel. Prokaryotic cells are usually small, simple. They have no organelles other than ribosomes. They first appeared 3.5 billion years ago, and they're typically unicellular. On the other hand, the other category of cells are eukaryotes, eu meaning true and kary kernel. So these means true nucleus. They're usually much larger, 10 to 100 times larger. They're more complex. They have membrane-enclosed organelles, and they appeared 2.1 billion years ago. And they can be either unicellular or multicellular. So down below in your slide, you can see on the left-hand side we have a prokaryotic cell, and on the right-hand side we have a eukaryotic cell, and that looks like a plant cell because it has a cell wall. So prokaryotic cells can have different shapes, but they include organisms from two domains, domain bacteria and domain archaea. And both of these are small unicellular organisms. So here you see Spirillum voludens, and you see another one, Methanosarcina. So here are two types of prokaryotic cells. And then on the right-hand side, you see eukaryotic cells, and these are found in the domain Eukarya. And these include plant and animal cells, fungal cells, and protists. And here we see allium, this would be from onion, and then actually a human lung cell. All prokaryotes are relatively simple-celled organisms. And again, the two domains are bacteria and archaea. And remember, they first appeared 3.5 billion years ago, so they're much older than eukaryotic cells. So here you see the two types of cells, bacteria and archaeans. And I love this diagram of a typical bacterial cell. So notice that there's not much in the cytoplasm. Within the cytoplasm, the interior of the cell, you see little dots, and those little dots are ribosomes. And the purpose of a ribosome is to actually make proteins or synthesize proteins. Then in pink, you see a whole bunch of squiggly material, and that's the nucleoid region, which contains the coiled DNA, and it's just one circular chromosome. Then you have like little tiny blue threads running throughout, and those are plasmids. They're small rings of DNA. But other than that, that's about what's in the interior of a bacterial cell. Then what's more complex is the plasma membrane, which regulates the passage of things going in and out, a cell wall that's used for protection, and then it can have an additional cover, which is called a capsule. And it's usually a sticky outer coat, and it provides another layer of protection. There are also short hairs that can be found outside, which are called pili. And then there's or long thread-like projections, which are called flagellum. And if there's many of them, then it's flagella. Did you know that scientists estimate that only maybe 1 in 10 of the cells in your body are actually human? The rest are largely prokaryote cells. These good bacteria help us digest food, synthesize vitamins, and protect against disease. So how is it possible to have more bacterial cells than human cells? And does knowing this change your opinion about bacteria? I always think of us as like moving islands, and on our island a lot of bacteria live. So we are definitely made of more bacteria than we are our own human cells. Animal and plant cells are made out of eukaryotic cells that contain organelles. So remember, animal and plants are found in domain eukarya, and eukaryotic cells are at least tenfold bigger and more complex than prokaryotic cells. Eukaryotic cells also contain organelles, and these organelles are usually membrane bound and they carry out specific functions. Plants and animals have many organelles in common. So what are some of the organelles that are in common to both plants and animals? Well, both contain a nucleus, both contain mitochondria, both contain cellular membranes, both contain ribosomes. So they contain a lot of things that are the same, but they also contain things that are different. I love this diagram. This is the structure of an idealized animal cell. So at first, at the top of the cell, you see the nuclear envelope enclosing the nucleus. And within the nucleus, you see the nucleolus. And the nucleolus, its function is to produce ribosomal RNA, which produces the ribozymes. Then outside of the nucleus, in blue, you see the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The rough produces protein, and the smooth is used to synthesize lipids. Then I see mitochondria, and the mitochondria here help with cellular respiration, and they take food molecules like glucose and convert it into energy. And there's also like little dots throughout the cytoplasm, and that cytoplasm is that inner matrix between the plasma membrane and the nucleus. And these ribosomes are the site of protein synthesis. And then you see the Golgi apparatus in like a teal color. And the Golgi apparatus is like the mail system, the U.S. mail system, but within the cell. It takes the products of proteins and it ships them throughout the cell in those little vesicles that you see budding off. Then you see other little vesicles that are called lysozymes. They can be, and lysozymes are like little stomachs inside of animal cells. You're not going to find them in plant cells. And they actually help recycle or digest old worn out organelles, or they also help break down things that are coming into the cell. Then we see the cytoskeleton. The cytoskeleton is a network of fibers, and they maintain the cell's shape, and they also give it support. So this is what an idealized animal cell looks like. A plant cell looks very similar to an animal cell, but there are some major differences, and it's obvious. And you can see it listed in green. First of all, there's a cell wall, and you don't see cell walls in animal cells. If I had a cell wall around each of my cells, I'd be walking like a robot, right? I'd lose all of my flexibility. Then there's something called a chloroplast, and that's what's putting in the green into plants. These chloroplasts are like little green beads inside of a plant cell. And these beads contain pigments, and these pigments are the one that harvest light energy and convert it into sugar. And then lastly, you see this huge space, and it's called a centriole. And this centriole contains mainly water, but it can contain pigments and nutrients or other things. And you sort of know when it's full and when it's empty. When it's full, your plants look nice and perky, and when it's not full, your plants tend to wilt. So that's why we always need to make sure to water our plants. So those are the three things or four things that are different. First of all, plants have a cell wall, they have a chloroplast, and they have a central vacuole. And all of these are really important because that's what makes them different from animal cells. Animal and plant cells have unique organelles. So for example, animals only have lysosomes. Those are those little stomachs. And on the other hand, plant cells have chloroplast cell walls and central vacuoles. So that's how you can tell the difference between the two. So the question is where does a cell start and stop? Like what defines a cell? What are the boundaries? And if we were to look under the microscope, what would you be looking for to find that boundary? And what criteria would you use? So it's something that we have talked about in previous chapters. And what we talked about is that phospholipid membrane, and this is what makes the cell or plasma membrane. So notice I can use plasma membrane or cell membrane interchangeably. They both mean the same, and they're both made out of a phospholipid bilayer. So here you see the membrane in a plant cell, you see it in an animal cell, and you also see it in a bacterial cell on the left-hand side of your slide. Plasma membranes are made up of two layers of phospholipids and integrated proteins. So the phosphate head is actually water-loving or hydrophilic, and the two fatty acid tails are hydrophobic, water-hating. So the heads always face out and the tails face in. I love this diagram of the cell membrane because it shows you how complex it is. So there in brown, you see the phospholipid bilayer, and you see the heads facing out, whether it's to the exterior outside of the cell or to the interior of the cell where there's a lot of liquid, and the tails are facing inward. But embedded in the cell are a lot of proteins indicated by these purple structures and blue ones. And then attached to them are actually green molecules. And these green molecules represent carbohydrates. So the cell membrane just isn't a phospholipid bilayer, but it contains a lot of proteins and carbohydrates. So notice there's a brown substance that looks like a hexagon. That almost looks like my beloved glucose. And notice that it's going through a protein. So we can see that many of the proteins that we see at the plasma membrane are acting like little gateways into the cell, like they're doorways or channels into the cell. Imagine that you and a group of friends are going out on a Saturday night. At the first club, the place is packed, and there's even a line. At the second club, the room is almost empty. So which club is more likely to have a cover charge, and which one is going to be harder to get into? We already know the answer. The more packed club is going to be really difficult to get into, and the one that's empty, it's going to be easy to get into. Now, imagine that you're a molecule trying to get into a cell across the cell membrane. Is it sort of like the clubs that we just talked about? And the answer is definitely yes. So we know that cells are surrounded by a cell or plasma membrane, and there's organelles in the inner part of the cell. And so if there's passage, then materials have to pass the plasma membrane to get into the inner part of the cell. So the plasma membrane regulates the flow of materials. So this flow of materials, some materials can get in and some can't. And this is called selective permeability. That is, some molecules can freely move in and some cannot. So there's a go, a stop, or yield. You know what this reminds me of? It sort of reminds me of a gated community. Some of the cars get to go in, and some of the cars are kept outside. So selective permeability. The other thing that we need to consider when materials are going across a membrane is it passive transport or active transport? Passive transport requires no energy. In other words, substances move along a concentration gradient. They move from a high concentration across a membrane to a low concentration. Active transport is just the opposite. It requires energy. And you know how I remember this? A is for ATP, A is for active. So active transport needs ATP, which is energy in the cell. So it moves against a concentration gradient. It moves from a low concentration to a high. You know what that reminds me of? It's like going in, trying to walk into a crowded club at nighttime. That is active transport. It'll be very hard to move inside. The other way I look at it too is passive transport to me is like having a bicycle and you're at the top of the Franklin Mountains. You're at the top and you just get on your bike and you don't even have to pedal down. You're going from a high to a low level. From a high concentration to a low level. It doesn't take any energy. You don't have to pedal. But active transport's the opposite. It's like being at the base of the mountain. So you have to go from the bottom all the way to the top. And you're going to have to pedal a lot. It requires energy. So that would be another way of looking at it. So when we say that molecules move across a membrane, the more appropriate word is diffusion, especially if it's passive transport. Remember, passive means it requires no outside energy. It moves from a high concentration to a low molecular concentration. This is called diffusion. So diffusion is the movement of molecules from an area of high concentration to an area of lower concentration. Now, there's a special word for water because water is so important. So the diffusion of water is called osmosis. So oftentimes the question is why does water move and not the molecules? Sometimes the molecules are too large or they have a charge and they can't move. And so to reach equilibrium, water needs to move across the cell membrane. And this is called osmosis. Water will always flow from an area of higher water concentration to an area of lower water concentration. I have a simple rule, like to remember, water always follows the solutes. Water always follows the salts. So whatever is dissolved in water, that's what water is going to follow. Okay, so water always follows the salts, and that way you know which way water is moving. Now, in these examples, we've seen that molecules are moving through a protein. So if they're moving through a protein that is actually embedded in the plasma membrane, then this is called facilitated diffusion. What does facilitate mean? Facilitate simply means to help. So if a protein is there, it's like a doorway into the cell. So it's helping move molecules through a doorway. So it's called facilitated diffusion. If molecules simply move between the phospholipids, then it doesn't need any protein help. So when proteins are involved, it's called facilitated diffusion. And remember that substances still move from an area of high concentration to an area of lower concentration. Active transport. Active transport requires energy in the form of ATP, and they all begin with the letter A. Active transport involves moving a substance from an area of lower concentration to an area of higher concentration. That's like going into a crowded club. Or it could be like me asking a student, please get on a bus where everyone's coming down from the bus, and I have to push him onto the bus. In other words, it requires energy. It's moving a substance against its concentration gradient, and because of this, it requires energy. One of the most classic examples of this is the sodium-potassium pump. But you know what the pump truly is? It's a protein that's embedded into the plasma membrane. And the protein is dependent on energy for it to move sodium, Na ions, and potassium, K ions, in opposite directions. So isn't it true that there's an abundance of sodium? So your cells don't want more sodium. They're always trying to pump it out. But outside of your cell is more sodium. So it's moving against a concentration gradient. And then on the opposite is potassium. So potassium isn't very common outside of the cell. Your cell keeps the potassium inside, but your cell always wants more. So it's moving also potassium against its concentration gradient. And because of this, it requires energy. So in this case, when we talk about pump, what are we talking about? We're talking about a protein embedded in the phospholipid bilayer. In my head, sometimes I think, I think of an old-fashioned bicycle pump where you're actually pumping and it's taking energy. Cells can also transport substances by fusing a portion of the cell membrane. So the cell membrane actually helps bring molecules into the cell. It can also help to take them outside of the cell. And it does this by forming these bubbles or vesicles. To me, a vesicle is just a fancy name for container. So it pinches off and it forms these like little containers. And I see them as little shipping containers. So here we have endocytosis and exocytosis. Exo means going out. Endo means going in. So cyte means cell. So exocytosis are things are leaving the cell. Endocytosis, things are being imported into the cell. Where did you get your chromosomes from? Okay, we know that humans have 46 chromosomes. So what are at least 3 things that you know about chromosomes? Could you draw one? Do you know what they're made of? Okay, so we'll be talking about that next when we talk about what is inside a cell. Okay, so first of all, where do you find chromosomes? We're going to find them in eukaryotic cells inside of the nucleus. So the nucleus is like the downtown of a cell. It's the heart of the cell. And this is where it's keeping its most important stuff. That's DNA. That's this information that makes us us. So it is membrane bound. And in every eukaryotic cell, you're going to see a nucleus. And so this includes both animal and plant cells. The nucleus contains most of the cell's DNA stored, and it's stored in the form of chromosomes. So the nucleus is surrounded by an envelope, and it contains DNA in the inside. And that's like the most important things that make us us. It's like our credit cards, our passport, our driver's license make us us, and we protect them. We just don't give them out to anyone. So we store them in a good place, and that's what the nucleus does. It's storing chromosomes in a safe place. The nucleus is surrounded by a double membrane called the nuclear envelope. So this is an up-close look of the nuclear envelope, and you can see that it has 2 membranes. And the other thing that you can see is that it has a lot of these like little openings or little like divots inside. So it has like these little pores, and in these pores, materials can travel in and out of the nucleus. The other thing that you see is another sphere inside of the nucleus, and that is called the nucleolus. And that nucleolus is what manufactures ribosomal RNA, which is another name for ribosomes. The other thing that you see inside of the nucleus is white stringy stuff. And that white stringy stuff is in fact the chromosomes themselves. And that's what they look like when they're not condensed. The nucleus houses chromosomes. It's that white stuff that we saw in the previous slide. But oftentimes when we look at a poster, we see that it looks like that iconic X. So how is it that it can go from stringy to looking like the X that we see on a lot of posters advertising biology? Well, it's called chromatin. The DNA, that stringy white stuff, starts wrapping around protein fibers called histones, and when it spools around the protein fibers, it condenses and it starts looking like that iconic X. So we can take long fibers and condense them down, and that looks like a condensed chromosome. The nucleolus is where ribosomal RNA is manufactured, but the truth is that ribosomes are made of ribosomal RNA. So this is the home place where ribosomes are made, and as soon as they're manufactured, they exit the nucleus and go into the cytoplasm. Who's in charge of the cell? Think of the cell as analogous to a factory. However, instead of producing widgets, the cellular factory produces proteins. So what roles might the following organelles play in the cell factory? So what's the role of the plasma membrane, the nucleus, ribosomes, and mitochondria? The DNA directs a cell's activity through the production of proteins. This is called central dogma. DNA, RNA, protein. I often say that we are our own 3D printer. We start with code. That's the code found on DNA. Then samples of that code are copied. That's RNA. And that RNA is taken out to the cytoplasm, to the ribosomes, where it's converted or translated into a protein. DNA in the nucleus contains the instructions for making proteins. And proteins make up most of the cell, and they make up most of us. Proteins are very important molecules in our cells, and they are involved in virtually all cell functions. So several organelles are involved in protein manufacturing. So in addition to DNA and RNA, we see that the endoplasmic reticulum is involved in protein production, and we also see that the Golgi apparatus is involved. So ribosomes, endoplasmic reticulum, and Golgi apparatus. So protein production involves two steps. The first step is transcription. So transcription is the copy of DNA to RNA, and that happens in the nucleus. And once that happens, the RNA leaves the nucleus and goes out into the cytoplasm where the ribosomes are located. Then at the ribosomes, the information on RNA is translated into a protein. And then from there, the Golgi apparatus will transport that protein to where it is needed in the cell. So translation occurs at the ribosomes. After transcription, RNA travels from the nucleus to a ribosome. Ribosomes are where proteins are made, and some ribosomes are bound to the membrane of the endoplasmic reticulum, or ER, and other ribosomes are free. So they're floating freely in the cytoplasm, and then they'll become attached to the endoplasmic reticulum. The smooth ER contains enzymes that produce lipids, and rough ER contains ribosomes that produce proteins. So actually the rough ER is where protein manufacturing takes place. The Golgi apparatus finishes, sorts, and ships cell products. So that's sort of, just like the postal service that we have in any city. It takes our mail or our packages, it sorts them, it puts labels on them, and sends them to the appropriate place. And so does the Golgi apparatus in the cell. Lysosomes. So what is the purpose of lysosomes? Well, lysosomes are like little stomachs, and they recycle worn out organelles, or they can dissolve large food molecules. So in this diagram here, you see a lysosome attaching to a worn out mitochondrion. And what it'll do is it will break it down, digest it, and break it down to building blocks that will be used for materials to build something else within the cell. What would happen if all the ribosomes in your cell disappeared? What would happen if half the mitochondria in your cells disappeared? What would happen? Do you think the cells would function? In my mind, it didn't take more than a split second to say the cell would definitely perish. Or in some cases, like with few mitochondria, then it would have to rely on other cells, and maybe it would become parasitic. Two organelles that help provide energy in the cell are chloroplasts, which are found in plant cells, and mitochondria, which are found in both plant cells and animal cells. So only chloroplasts are found in plant cells, but keep in mind, mitochondria are found in both plant and animal cells. So chloroplasts are the organelle of photosynthesis. So photosynthesis means light making, and we know that light in this case will make sugar. And so the pigments inside of the chloroplasts are used to capture the energy from the sun and to make sugar from a supply of carbon dioxide and water. Sugars produced by photosynthesis provide the energy to power the cell. And when we eat those sugars, it powers our cells. So here's a chloroplast. Chloro means green and plast means body. Chloroplast, green body. And that definitely is what's making plant cells look green. So within the chloroplast, we see that there's an inner and outer membrane, and within we see stacks of thylakoids. And these stacks contain the pigments that will be used to harvest light to have enough energy to make sugar from CO2. In the mitochondria, that's another special organelle, and it is used for cellular respiration. So in this case, cellular respiration uses oxygen to harvest energy from molecules of sugar. So this energy then is converted into ATP, and this is what powers the cell and gives energy for all of the chemical reactions. So here we see a mitochondrion, and notice it too has an inner and outer membrane. And this is where sugar, in the presence of oxygen, is converted to energy in the form of ATP. And at the same time, you can see it releases CO2 and water. What are three structures in the plant that are not found in animal cells? Do you remember what I listed before? For each of these structures, explain why it's useful for plant cells but not for animal cells. Okay, so remember, one of the first structures that is unique to a plant cell is a vacuole. So vacuoles are intercellular sacs, and they're for storage of food and nutrients or pigments, but oftentimes they contain water. Many plant cells have very large central vacuoles. They're containing a lot of water. Some cells have moving appendages, like flagella. Okay, so what are flagella? They're long hair-like structures. And in this diagram, we see human sperm. And each of these, human sperm to me looks like a little Christmas light with a long tail on it. So we can see that it is moving the sperm along, the flagella. And some of our cells actually have cilia. So if you look in your throat, you see that actually the windpipe area, the cells have cilia. They're little tiny hair-like appendages, and they move back and forth, and they remove a lot of that phlegm or mucus that's in our throat. So when you start smoking, then the cilia stop moving back and forth, and that's why you have mucus accumulate in your throat. Some cells are supported by a rigid cell wall surrounding the membrane. So another unique structure is the cell wall. And this is found in plant cells, but it is not found in animal cells. So notice here you have some diagrams of an animal cell, and then you also have one of an algal cell. Animal cells maintain their shape with an internal cytoskeleton. And a cytoskeleton is a network of protein fibers that provides mechanical support, anchorage, and reinforcement. The cytoskeleton network can be quickly dismantled and reassembled. So that's what gives the cell its shape, or all of these protein fibers. Animals also have an extracellular matrix. They may not have a cell wall, but they do have an extracellular matrix, and that helps hold cells together, and it also helps signal and recognize other cells. So cells are held together into a tissue by an extracellular matrix. The rest of this chapter has to do with the appendix of definitions, and you can find this in the regular PowerPoint presentation. So this is the end of chapter 3.